Hello, my lovely students. So, here I am to do the recap of the chapter Plant Growth and Development. This is the last chapter of your 11th standard. So, without any delay, let's start with our chapter. So, in plant growth development, all of you know that we talk about plant growth regulators, right? What are they? They are small molecules which help in either plant growth, for promoting plant growth, or for inhibiting them, right? So there are two categories of plant growth regulators. First is plant growth promoters, right? They are auxin, gibberellin, and cytokinin. So these three plant growth regulators are basically promoting the growth, whereas growth growth inhibitors are abscisic acid, right? It inhibits the growth. So whenever any stressful condition is there, this abscisic acid comes in the rescue and rescue the plant from any stressful condition, right? So you must be wondering, where is ethylene? So ethylene basically is a type of hormone which can be fit in both the categories. Either it, in some cases they can promote the growth, and in some cases it can inhibit the growth. So ethylene is basically growth inhibiting hormone. Okay, ethylene is basically growth inhibiting hormone. It inhibits the growth of plant, right? It can or it can be growth promoting hormone, right? So ethylene can be fit in either of these category. Either it can promote the growth or it can inhibit the growth. So in this revision, we are going to revise the functions and the discovery of these hormones, and we will start first with auxin, right? Remember, there are two type of auxin: natural, which is naturally found in the plant, and artificial. Natural and artificial. Natural auxin are indole acetic acid and your indole butyric acid. IAA is indole acetic acid, IBA is indole butyric acid. In artificial is 2,4 dichlorophenoxyacetic acid and 2,4,5 2,4,5 trichlorophenoliacetic acid. So these are some of the artificial auxin which is not naturally present in the plant. Okay, so let's start with the discovery of auxin. So auxin discovery, basically its discovery started with the Charles Darwin and his son. He observed that the tip of canary grass plant responded to unilateral illumination by growing towards the light. So what they observed, that when the light is provided from one side, the shoot grew towards the light, right? So this is what they found, when where from wherever light is coming, the shoot will bend towards it. Then FW went was the first one to isolate or discovered auxin. He isolated from oat seedling. Oat is a plant, and from its seedling, FW went first isolated this hormone. So this was the first hormone to be discovered, and its discovery credit is given to FW Went. Let's discuss its functions. It has many function. One of the function is it promotes rooting, right? Whenever you apply auxin, it promotes the root formation. It promotes apical dominance. What is apical dominance, beta? Apical dominance is, this is apical bud. The bud is the tip. It has high concentration of auxin. It has high concentration of auxin. So whenever this apical bud is present, it won't allow this lateral bud to grow. This is lateral bud. Okay, so here this is known as apical dominance. So whenever apical bud is there, it won't allow lateral bud to grow. This is known as apical dominance. It initiates flowering in pineapple. Of course, it initiates flowering in pineapple plant, and it prevents drooping of fruit and flower. Yes, it prevents the drooping of fruit and flower. When we remove the shoot tip, when you remove the shoot tip, then it can lead to the growth of lateral buds. More lateral buds means more leaf. And what do we need in tea plant? We need leaf. So more branches means more leaf, and more leaf means more production. So in tea plantation, what does the farmers do? They just cut the tip so that lateral buds can grow and more leaves can be produced. Okay. Then it can use in parthenocarpy. As you know, parthenocarpy is the formation of fruit without fertilization. So whenever the ovary is directly developing into a fruit without any fertilization, then there will be high concentration of auxin in the plant, in the ovary, so that ovary can directly develop into fruit without fertilization. It is used as a herbicide, especially 2,4-D auxin, which is an artificial auxin. It can be used as a herbicide. Herbicide kills unwanted plant. Herbicides kill unwanted plants. Okay, so these are the some of the functions of auxin. Very important, right? Very important. So I'm going to give you 10 seconds. 10 seconds to look at each point. 10 seconds to look at each point, and then let me know if you have any doubt, if you have any doubt in any of the point. Okay, let me know in the comment. 5 seconds. Okay, next is gibberellin, which is also known as gibberellic acid. Okay, so what happened is, in Japan, some farmer observed that the rice plant grew excessively tall, right? So they called it buccane or foolish disease. Later on, some pathologists came and they found that this disease was caused by a fungus known as Gibberella fujikuroi. So it means gibberellin, the source is fungi and plant. Okay, so gibberellin can be extracted from either fungi or plants, and the first gibberellin was GA3, was the first gibberellin to be discovered. There are many gibberellins, more than 100 gibberellins discovered so far, but gibberellic acid 3 was the first one to be discovered among gibberellins. Okay. So, who was the scientist? Who was the pathologist? E. Kurosawa reported the appearance of symptoms of the disease in unaffected rice when they were treated with the fungus. So, what did E. Kurosawa do? He transferred the infection from unhealthy to healthy plant, and healthy plants also developed the symptoms. So, he found out that it's a fungal infection. Okay. Let's see what are the functions of this gibberellin. How can we use this gibberellic acid in our own plant production? So they increase the length of grape stalk. So more the length of grape stalk, more will be the number of grapes. Gibberellin causes fruit like apple to elongate and improve its shape. The beautiful shape of apple that you see is mostly because of gibberellin. Gibberellin tries to improve the shape of the apple. It delays senescence. Senescence means aging. Senescence means aging, which means that gibberellin delays the aging. GA3 is used to speed up the malting process in brewing industry. Okay, so what are malt? Malt is the barley seed or rye seed or wheat seed, which is basically which is germinated and then fermented. So this process is known as malting process, and GA3 or gibberellic acid 3 is added to speed up this process so that uh, in a very less time the alcoholic beverages could be produced. Now, the spraying sugar cane crop with gibberellin increases the length of the stem, thus increasing the yield by 20 tons per acre. So as you know, the part that we utilize in sugar cane is a stem. More stem means more sugar, right? More sugar means more yield. So yes, this gibberellic acid is used to elongate the length of the sugar cane, right? 10 seconds here. Okay. Next is here gibberellic acid. Gibberellic acid, another function is spraying juvenile. Juvenile means young. And conifers are gymnosperms. So spraying young conifers with gibberellic acid speed up the maturity period, thus leading to early seed production, right? So if we spray gibberellic acid to the young gymnosperm, to the young conifer, we will get seed in a very less time. Okay. And gibberellic acid also promotes bolting in beet, cabbage. Bolting means when a dwarf plant, okay, this is a plant with rosette habit. As you know, the plant of cabbage, beet are very small, so that we call it rosette habit, very small plant. Okay, so plant with rosette habit such as cabbage, beet, if we spray gibberellic acid just before the flowering, the length of the stem increases. The length of the stem increases. This process is known as bolting. This process is known as bolting. So earlier the plant was dwarf. Earlier, earlier the plant was dwarf, but now the stem has been elongated. The stem has been elongated, right? So this is the last function of gibberellic acid. This is the last function of gibberellic acid. After this, we will see cytokinin. We will see the third hormone known as cytokinin. Okay, cytokinin, right? Let's see from where its discovery started. F. Cook and his co-worker, they observed that the internodal segment of tobacco, so this is the callus from tobacco plant. Callus means the mass of cell. Callus means mass of cell. Okay, it only proliferate. It only proliferate when auxin plus cytokinin were given. Okay, proliferate means to make stem and root. Proliferate means to form stem and root. So what they are saying that F. Cook found out that this mass of cell from tobacco only made the stem and root when it was supplied with auxin as well as cytokinin. What is the source of cytokinin? Either vascular tissue, yeast extract, milk, coconut milk, or DNA. It's a source of cytokinin. It's a source of cytokinin. Okay. Then Skoog and Miller identify the first cytokinin. Remember the natural cytokinin. The name of natural cytokinin is zeatin. It was discovered from maize plant, right? And the name of artificial cytokinin, it was discovered from the fish, kinetin. Another name of kinetin is N6 furfuryl aminopurin. Another name of kinetin is N6 furfuryl aminopurin. Kinetin is basically the artificial cytokinin. Okay. Let's see the function of cytokinin. As all of you know that cytokinin are basically synthesized in the region where very rapid cell division is occurring. All the young part are having high concentration of cytokinin. It helps to produce new leaves, new chloroplast in the leaf, and lateral shoot growth. Lateral bud, right? Growth. So cytokinin is doing opposite of auxin. Auxin is inhibiting the lateral growth, whereas cytokinin is promoting the lateral growth. So cytokinin help in overcoming the apical dominance. It basically promotes the growth of lateral bud, whereas auxin inhibits the growth of lateral buds. They promote nutrient mobilization, which help in the delay of senescence, which means movement of nutrients to young part, right? So cytokinin makes sure that the nutrients will move to the young part. The nutrients will move to the young part. Is that clear to all of you? Please do let me know in the comment section. After this, the second last hormone, which is ethylene. Second last hormone, ethylene. 10 seconds here, 10 seconds here. Then we'll see ethylene. What does ethylene do? It had lots and lots of functions. Okay. Next is ethylene. So discovery of ethylene starts with cousins. So cousins confirmed that when you put orange with banana, there is some sort of chemical, gaseous chemical releasing from orange, which is ripening the banana, right? So the release of volatile substance from the oranges that hasten the ripening of unripened banana. And later on, this substance was discovered to be ethylene. So whenever you put oranges with banana, the oranges led to the ripening of banana in a very faster speed. Okay. So what does this ethylene do? It promotes senescence. Senescence means aging. It promotes abscission. Abscission means falling down of leaf, falling down of leaves or other parts. This is known as abscission. It promotes that. It enhances the ripening of fruits. Yes, ethylene promotes the ripening of fruit. It breaks the seed and bud dormancy. Beta, dormancy means resting phase. Dormancy means resting phase. So it makes sure that the seed and bud comes out of this resting phase. So it breaks the dormancy so that bud and seed can come out of the resting phase. It initiates germination in peanut seed and also sprouting of potato tubers. So many times you may have observed in your kitchen that the potato starts sprouting, right? You can see the white green structure over the potato, and that can be because of high concentration of ethylene. They cause very rapid elongation in the deep water rice plant. So they make sure that in the deep water rice plant, the rice plant comes out of the water, right? So rice water should not be submerged in the water, it should be come out of the water. That is achieved with the help of ethylene. It also promotes root formation and root hair formation, and it also initiates flowering in pineapple and mango. Okay, so there are more functions. I will give you some time here, 10 seconds. 10 seconds here. Observe each one of the function here. 5 seconds. Okay, let's see the next function. Ethaphone is an artificial ethylene. It's an aqueous solution. When we spray this to plant, it releases ethylene. So ethaphone is an artificial solution of ethylene. Ethaphone speed up the ripening of tomatoes, apples, and accelerate abscission in flower and fruit. It makes sure that the flower and fruit, when get mature, it falls down. Thinning of cotton and cherry and walnut. So ethylene makes sure there will be thinning of the fruit wall of walnut, cherry, and cotton. It promotes the female flower in cucumber plant, which means more female flower means more fruit. More female flowers means more fruits. Okay, so these are the roles of ethylene. These are the roles of ethylene. Okay, these are the roles of ethylene. I hope this is clear to all of you. So let's move on to the last hormone, which is abscisic acid. Okay, last hormone, which is abscisic acid. Abscisic acid is also known as stress hormone. It is also known as stress hormone. It makes sure to close the stomata, closure of stomata during water scarcity. During water scarcity. So when there is a less water, it makes sure that the stomata will be closed. It promotes seed dormancy in case of unfavorable condition. So whenever there is an unfavorable condition, it makes sure that the seed won't germinate unless the seed will get the favorable condition, unless the seed will get the favorable condition. So it makes sure that it promotes seed dormancy. It makes sure that it promotes seed dormancy, right? So that's why the ABA is known as stress hormone. ABA known as stress hormone. Okay, let's solve some questions. Let's solve some questions. The growth hormone auxin isolated from FW went from the tips of seedling. All of you know this answer, oats. So FW went basically extracted the auxin first from the oats plant. First from the oats plant, right? First from the oats plant. I hope this is clear to all of you. Sorry. The next is fruit and leaf drop at early stage can be prevented by the application of. So at early stage, the leaf and the fruit droop can be prevented by auxin. Can be prevented by auxin. Can be prevented by auxin. Okay. Next is, it takes a long time for pineapple plant to produce flower. Which combination of hormone can be applied to artificially induce flowering in pineapple plant throughout the year to increase yield? Right? So it is auxin and ethylene. We have to apply the combination of auxin and ethylene to promote flowers or flowering in pineapple plant. Okay? Last question, match the following. The fruit ripener is ethaphone. The herbicide is 2,4-D, which is auxin. The bolting agent is gibberellic acid. And the stress hormone is abscisic acid. Stress hormone is abscisic acid. Very easy, match the following, right? I hope all the topics and questions are clear to you. So this was the last chapter of the 11th standard plant growth and development, and I really hope every topic is very clear to you. So make sure you read every point and line from the NCERT, and do let me know in the comment section if you have any doubt. So we'll see you in the later on next time. Till then, take care everyone. Bye.